Hello students, this is a recording for Biology 1407, Unit 3, Chapter 33 in the OpenStax textbook, Animal Form and Function. So again, the subject matter is animal form and function. I'm going to go through the chapter in the textbook using the online textbook. I also want to remind you of the study guide that is posted, and I'll refer to this as well. It has some slightly extended objectives in here, and also a video which I encourage you to watch, which primarily talks about tissues and how the tissues are arranged in animals. And then we'll go through these items, which are covered in the textbook. I'll add a little bit more than the textbook says to fully answer these statements right here, and then we'll work our way through the material. All right. So this is chapter 33 in your textbook. All right. So we start by remembering body plans, and you'll recall that all of the animals that we've looked at are either going to be asymmetrical. This will be sponges, poriferans, exhibit radial symmetry, which is the cnidarians or the ctenophorans, and the bulk of the rest of the animals which have bilateral symmetry, although a few of them have secondary radial symmetry. Think about a sea urchin or a starfish or something like that. But embryonically and critically in internal anatomy, they are bilateral, like this animal right here. So there's a left and a right side, an anterior and a posterior, an inferior and a superior, or ventral and dorsal, depending on what kind of animal you're talking about. All right, so we don't really need to go too much back over those shapes, just remember them, please. Limits on animal size and shape. This is an important idea right here. So for instance, it's talking about animals that live in water, and one of the convergent evolutionary things that we see is that all animals that swim and need to swim quickly tend to take on or be able to induce a fusiform shape. So that's the shape, that's the proper term for the shape of a streamlined fish, that one that's built for speed. Don't think about some sort of coral reef fish that barely moves around and has spikes and things sticking off of it all over. But think about a shark, your basic shark. That's an excellent example of fusiform. Or a tuna fish, or a penguin, or a squid when it's propelling itself in jet propulsion. Or a lobster when it's shooting itself backwards using its tail to move itself, or a shrimp. They all end up having roughly the same sort of shape, which is rounded, a little bit more bulbous at the head end, tapering off like a teardrop posteriorly, and that's the shape that allows an animal to move quickly through the water. So that puts a limitation, on the size and shapes of organisms in the water if they're going to pursue things at high speeds. They all have to tend to this fusiform shape. They can get quite large, but think about a blue whale, perhaps the biggest animal that's ever lived on Earth by full dimensions and weight, by its bulk particularly. But that is a restraining feature. And so that's kind of the point right here. The environment dictates what it is possible for an animal to achieve as far as shape, function, speed, places it can live and all of that, the environment. So on land, animals, on the other hand, are not really concerned with drag in the water. It's gravity, as it says right here, that becomes the most important limiting factor. And so the way that works is, as animals get larger, things change in an exponential way. So if an animal is twice as big, it's not going to have twice as much skeletal mass. It'll actually have to have something more than that. Perhaps not the square power, but the 1.5 power, or the 1 3/4 power, or something like that. So as animals get bigger and bigger, their skeletons become a larger and larger part of the entire animal, and then the musculature required to move that bigger and heavier skeleton gets bigger too. So when you look at a mouse, of course it's small and not super fast compared to some larger animals, but it has an incredibly lightweight, delicate skeleton and relatively small musculature. The ratio of muscles to other organs in the body is relatively low. When you get up to an elephant, think of an elephant walking around, the amount of the body weight that's dedicated to the skeleton and the skeletal muscles is a much larger percentage of the total weight of the animal. And that kind of limits how big animals can get. So on land, there were some dinosaurs that were substantially larger than elephants, but, not too many varieties, and most of the dinosaurs also would have been elephant-sized or smaller. And I'm thinking of the big ones. The bulk of dinosaurs would have all been small animals, the size of a dog or a small bird or a rat or something like that. It's just the big spectacular ones that kind of populate our imagination. But the reason they couldn't get as large as, say, a blue whale in the water is because of the force of gravity requires so much skeletal and muscular mass that an animal over a certain size could never get enough energy intake in order to keep all of that muscle alive. So that's kind of a limit on their size. And of course, elephants can trot along quite quickly. They sort of gamble along pretty quickly, but they can't run like a horse can because of the large size. So they're constrained to, a slower relative speed, compared to their body size than a smaller animal would be capable of. So this is kind of the idea of limits on animal size and shape. Same thing for flying animals. As birds get bigger and bigger, the amount of surface area that has to be devoted to wings increases more rapidly than the linear dimensions of the bird. So really large birds have terrifically huge wings compared to their body size, compared to a really small bird like a hummingbird or a sparrow or something like that. Same sort of limitation. So you can't have an infinitely large skeletal animal wandering around on land, nor can you have an infinitely large bird flying around. There's limits. Also, interestingly, in the world of skeletons, an exoskeleton, as we're seeing right here, that you find on arthropods, does work on land, but as the animal gets bigger and bigger, the thickness and weight of the exoskeleton becomes prohibitive even more rapidly than is true in an animal with an endoskeleton like us, with the skeleton on the inside. And really the weight of the skeleton is the limit on how big an arthropod can be on land. And so that constrains them to sizes quite a bit smaller than vertebrates with endoskeletons are capable of. That's why the vertebrate skeleton is such an interesting innovation in the world of animals, because it allows for substantially larger size on land, but not without limit. And that's really the whole point. So another idea that really limits and defines how animals are put together is the effect of diffusion on size and development. So if you look at a single cell, as you increase the radius of that cell, what happens is the surface area increases as a square of the radius. Surface area, 4 pi r squared right here, while the volume increases as a cube of the radius, 4/3 pi r cubed right here. What does that translate into? Well, as the cell gets bigger and bigger, there's more stuff inside that has to be supplied by not as much surface area, membrane, for each unit of inside matter. So what this means is, as cells get larger and larger, their ability to exchange materials with the outside world, oxygen in, let's say, CO2 out, nutrients in, waste products like ammonia out, all of that becomes harder and harder as the cell gets larger and larger. And finally you reach a size where that doesn't work anymore, and that kind of defines the maximum size of an animal cell. So if you're not aware of this, I'll tell you right now, the typical cell in a blue whale is the same size as a typical cell in a shrew or a mouse. The bigger animal simply has more cells, of course, but the individual cells can't increase beyond a certain linear size and therefore surface to volume ratio. And that's a huge limitation right there. So if you want to have a multicellular animal, what you have to do is A, chop that animal into small cells, and those cells have to be exposed to the medium that they're going to exchange materials with. So that's the outside world. So, as animals get larger, they tend to stop being spherical, which has the smallest surface area to volume ratio, and instead tend to be more elongated, flattened, thinned down to increase the amount of surface area that is exposed to the outside world relative to the full volume of cells inside the animal. And then to increase in size beyond that, what you'll find is that animals start having body cavities, which allows for more surface area on the inside. So if you wash some fluid along those internal surface areas, you have more potential for exchange of gases, exchange for nutrients, exchange for waste products, hormones, and all of that sort of stuff. And there is a limit. So as that animal gets bigger and bigger, what you've got to do is keep the distance between the individual cells making up that animal and some surface that's connected to the outside world down within a certain limit. And if you look at the study guide, I quote, this is a rule of thumb. There's lots of variation to this idea right here. But that typically a cell needs to be no more than about 6 cells away from a surface where diffusion can occur. So you can't have an animal that's 20 cells thick, or 40 cells thick, or 80 cells thick without having discontinuities or breaks inside there where we have a surface for exchange of materials. And what we find is inevitably that's the reason we have body cavities in larger bilateral animals. And then very, very quickly we start seeing circulatory systems start showing up. Initially they're relatively passive. Fluid is pumped around by movement of the animal, but then inevitably we start seeing hearts and various other pumping devices to move this liquid around. And that is to reduce that diffusion distance so that each cell is, virtually speaking, not more than about 6 cells away from a source of exchangeable nutrients. Alrighty, let's go back to the chapter right here. So I hope that gives you some understanding of this problem with diffusion right here. So diffusion distances become impossible to support if a cell gets larger than your typical cell of about 100 micrometers in diameter, so 50 micrometers in radius or so. And for a multicellular organism, if those cells are more than about 6 cells away from a surface, either an internal exchange surface or an external surface where they can exchange materials with. So remember that rule, that 6-cell rule, and why that makes sense. Another thing about energetics. So here's an interesting thing that, so gravity and drag and diffusion tend to reward animals for being smaller, right? But competition with other things, the tendency to be eaten by other things, definitely rewards animals for being larger. So that's one of the dynamics at work, that determines the size of an animal. Efficiency is really, efficiency on those scaling issues, the ability to fly, the amount of skeletal and muscular tissue that's required to move you around. All those things work better if you're smaller, the efficiency of a skeleton. But there's a lot of good reasons from a competitive world, from an ecological world, to be large. Another physiological reason that animals tend to be larger is because of energetics, what's called metabolic rate right here. And this is particularly true for warm-blooded animals, less so for cold-blooded animals. But so if we're looking at warm-blooded animals, which are referred to as endotherms, meaning their temperature comes from inside or they retain their temperature inside, versus ectotherms, whose body temperature is dependent on their environment, you'll find that endotherms actually work more efficiently as they get larger and larger. So what they're showing right here is the difference between a shrew or a mouse. What is this? Is some sort of a mouse here in this particular case, and an elephant again. All right, and so the mouse is about 35 grams, and the elephant over here is about 4.5 million grams, or 4.5 tons, metric tons, as it turns out right there. A pretty big animal. And the metabolic rate is, you see right here, 890 millimeters squared, so cubic millimeters of oxygen per gram of body per hour. So for a unit, a standard unit, 1 gram of body mass, it's going to need 800 cubic milliliters of oxygen per gram per hour. Whereas this very large animal right here only needs 75 millimeters of oxygen, cubic millimeters of oxygen per gram per hour. Most of the energy that this animal is burning, and the oxygen is an easy way of measuring how much cellular respiration, aerobic cellular respiration, is occurring in an animal. This animal needs to do quite a bit more aerobic respiration in order to generate body heat. A huge amount of energy is going just to maintain body heat. Whereas in this animal right here, a much smaller amount of energy is being devoted to generating body heat. About a tenth of the amount per gram per hour. So this is a much more efficient animal at keeping itself warm. And the reason that works is because of size. The surface area to volume ratio of this animal favors surface area, so it loses heat very quickly and very efficiently to the environment. The elephant, on the other hand, has a much larger internal volume relative to its surface area. Now, a couple of other things going on here. This mouse has compensated somewhat by having small ears, a small, dense, round body, minimum amount of surface area for a given volume. It's heading towards being almost spherical in shape, so that makes it as efficient as it can be while being really small here, but still it's, very, very porous thermally to the outside world. Of course, it's covered with fur, a typical mammal endotherm adaptation to retain body heat, and it could even have a layer of subcutaneous fat to improve that heat retention as well, but it's still an expensive animal to run per gram. This elephant, on the other hand, has some adjustable huge surface area. The ears here are for heat release, so if it gets overheated, it'll transfer a lot of blood flow to the ears. Instead of being cool, these ears will be red hot, not red hot, but body hot, and radiating heat into their environment. So it can adjust a little bit too. This is an animal that's pretty obviously adapted to live in a warm environment. So there's a whole bunch of information when just considering basal metabolic rate. So what y'all need to remember is that a small animal inevitably has a higher, more expensive metabolism than a large animal per unit of weight, per standard unit. Right? Of course, this elephant needs more energy absolutely in food than this mouse. It's a much huger animal, but the amount of food it needs per gram is quite a bit less than the food this thing needs per gram. This guy will starve to death in a day or two, in a couple of days, without a steady supply of food, unless it's hibernating. Let's say, 2 or 3 days. Whereas this thing could last weeks, even a month without food, without too much ill effect if it's in good shape. All right, so that's a little bit about basal metabolism and energy consumption. All right. Also, another little idea right here, it talks about herbivores and herbivore foods right here. So as animals get larger and larger, the ability to successfully, regularly acquire food becomes a greater and greater challenge. So the largest animals tend not to be carnivores, but will all, inevitably be herbivores. That is a pretty standard transition that you'll see. The largest members of any group of animals will always be herbivores or some sort of filter feeders where they can bulk feed on stuff that's available all around them rather than having to expend huge amounts of energy chasing down prey. All right, a little bit more about the environment and temperature. So, there are several different ways in which animals can, adapt to temperature and differences in temperature. And one of those is what we call hibernation, which is typically more of a torpor thing, although there are some things that go into a true hibernation. So torpor is simply slowing down activity, metabolism, breathing, energy consumption. We do that when we go to sleep. Our basal metabolism drops substantially while we're asleep. It's still high. A big chunk of our metabolism is always devoted to just maintaining our temperature and basically just keeping the motor running, the body working without actually doing anything. Then you have to add on more about metabolic costs for getting up and walking or running or things like that. All right. But so animals with that can regulate their internal temperature, endotherms. Let's go back to wherever it was up here. Endotherms right here. If they have a food source, they can live in really, really challenging environments. Think of all of the animals living in Antarctica or at the North Pole. Above water, you're not going to find any invertebrates or any cold-blooded animals. Everything is a vertebrate, and it is warm-blooded, birds and mammals. All right, and then underwater, a lot of fish, of course, and arthropods and all kinds of stuff. And the reason for that is temperature regulation. If a cold-blooded, if an ectothermic animal tried to live on the surface of Antarctica or on top of ice or on ice sheets in Greenland, northern Alaska, places like that, it would freeze to death. It would become frozen. The temperature is clearly below the freezing temperature of water. All right. When you go into the ocean, even the ocean under the ice at the North Pole, that water is just above freezing, because otherwise it would be ice. Some of that is a function of what's dissolved in the water. So the water is relatively salty. That depresses its freezing temperature. And the fluids of those animals are also very, very salty, full of sugars, full of other proteins. That decreases the freezing temperature, lowers the temperature the liquid has to get to before it freezes for those animals. But an animal can't exist if it can't keep from freezing. And there's two ways of doing that. Be in a place that's not freezing, and that's all animals living in liquid water. Or if you are in a place that is below freezing, you've got to be an endotherm. You have to have an internal source of heat. All right. Now we want to go over the tissues. This is true for all animals. So all animals are made out of these tissues in some combination. All right. And the cells that make up these tissues are very, very conserved and very, very similar across all animals out there. We're all built of the same sort of cellular building blocks, which are in turn made out of the same shared molecular building blocks, with a few special things for each group of animals. There's some toxins that a whole bunch of amphibians make that I don't make as a mammal. But, the things that are common, that are made, the molecules, including proteins, and therefore cells that are made out of those proteins and lipids and things like that, are more similar than they are different from animal to animal. How you organize them, you get the different shapes. All right. So, animals are composed of 4 different types of tissue. You should know that. There's epithelial tissue. There is muscular tissue. There is nervous tissue. And then there is this great big grab bag of tissues called connective tissues. All right. And you'll want to remember, and I'll go back to the study guide right here, there were the 3 embryonic tissues, you'll recall, from our study of animal development. And those 3 embryonic tissues are endotherm, endoderm, mesoderm, and ectoderm. So endoderm lines what's going to be the future internal lining of the digestive system. Mesoderm is going to be everything in the middle, between the digestive system and the outside of the body. And then ectoderm will cover external surfaces, including any folds leading in. All right. So any surface, that means, on the body, the external surface, or the surface of any tube or tubes or things in the body that lead, without having to cross the cell membrane, to the outside surface, is going to be derived from ectoderm. And that's what the epithelia are. That's what those epithelia are. Surfaces that lead to the outside world. So if you think about the lining of your lungs, if you go into your lungs, and you were somehow to be able to go into one of the tiny, tiny little alveoli, the gas exchange sacs, and be on that inside surface, you can follow the air path all the way out to the outside world. That surface, therefore, is referred to as an epithelium. It's derived from ectoderm. All right. So there's a lot right here. We distinguish the epithelia based on features, shape, and layers. All right, so we'll go ahead and take a look at that in the presentation right here. When we take a look at epithelia, the shapes, they can either be squamous, which is flattened, sort of pancake-shaped cells, cuboidal, name describes it, looks like a dice, roughly, an organic, blobby one. Columnar, like a tall column, literally, or something sort of in between those two. And then the other possibility is, besides the shapes, so here's an example of some squamous cells looked at from side. These are the cells flaking off the surface of your skin right here. Here's the transition between living cells in your skin and dead cells. And this is just the epithelium. This is the layer that comes off when you get sunburned. All right. Here's some cuboidal cells. They almost invariably surround a central canal. And if you see cuboidal tissue, you should think gland, like a digestive gland producing digestive liquids that go down this tube and into the stomach or into the intestines. Salivary glands, sweat glands, mammary glands, any kind of gland will be built of these sorts of cells right here. And they make hollow tubes whose contents lead to a surface somewhere, either a surface inside your body, or an external surface like the sweat glands all over my skin. Here's an example of columnar epithelium with cilia on top of it, which is typical of your airwaves. And this tissue right here, these are mucus-producing cells in your airways, produce a thick, mucus that lines the surface of your airways, and then these cilia pull that mucus continuously up, 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 out of your lungs and into your throat, where you can swallow or spit out that mucus. And everything with a respiratory system has the same sort of function going on in here. Every vertebrate with a respiratory system. And here's an example of some pseudostride ciliated columnar epithelium. All right. The other thing is, we don't go into a lot more detail of it right here, but the cells, if you have layers and layers of cells right here, that is typically meant to be a protective surface. So the outside of your skin is made out of many dozens of layers of epithelium, and that is to protect the underlying connective tissue and blood supply and everything from the outside world. You don't want any exchange of anything across this surface right here. And that's why you will have epithelial tissues that are made out of layers, lots and lots of layers. Where you want a whole bunch of exchange or secretion of materials, they'll typically have an epithelium just made out of one layer of cells, like this cuboidal epithelium right here, or this transitional, this columnar epithelium right here in the digestive tract or in the respiratory tract right here. All right, next, let's go ahead and take a look at connective tissue going through the book. In the study guide, it's actually the fourth item right here because it's got the most information. But we'll talk about this connective tissue a little bit. All right, so we can divide up connective tissue basically by whether it is loose or dense. And loose just means that there's a lot of room for liquid to move around in that connective tissue. So that's typically going to be connective tissue that you find around your capillary beds anywhere in your body. That'll be this areolar connective tissue. Adipose tissue is fat tissue that's kind of a modified version of that areolar. But these are very, very loose connective tissues that allow for materials to move through them quite easily. All right, and typically what we find around circulatory tissue, around a capillary bed. Dense fibrous is what we call tendons or the sheet, versions of connective tissue. So the regular dense fibrous connective tissue would be a tendon or a ligament. I think that's fairly self-explanatory. They're made out of lots and lots of fibers packed really close together. And the irregular fibrous connective tissues are the things that surround and hold an organ together. You have some of this wrapping around every muscle. It's surrounded by a layer of what's called irregular dense fibrous connective tissue that holds it all together. the dermis of your skin, the thick part of your skin, not the surface that's made out of epithelium, but the thick bit that I'm pinching and lifting right here. That is going to be a very thick layer of dense, irregular, fibrous connective tissue. That's the dermal layer of your skin. You have the epidermis, then you have the dermis, and then you have the hypodermis, which is adipose tissue connecting to the muscle down below, or to the connective tissue surrounding the muscle down below, I should say. So that's the way those guys work. And then, of course, two very specialized sorts of connective tissue, and this is where these are full of liquid. Lots of liquid in the case of loose fibrous connective tissue. Basically, the plasma of blood is just the liquid part of blood leaked out. In dense fibrous connective tissue, less liquid, because the fibers are really close together. And these fibers, by the way, are collagen. That's the primary fiber you're talking about in all animals. If there's a fibrous protein inside, it's going to be collagen. Well, the skeletal and blood, or skeletal and, let me just do the skeletal first. The skeletal tissue replaces that fluid primarily with either something solid, in the case of bone, that's calcium phosphate crystals, or cartilage, a tough gelatinous, hydrated gel-like material that makes up what was the liquid in these tissues right here, becomes the jelly-like part of cartilage or the hard crystalline part of bone. Skeletal connective tissue still has lots of fibers. There's a huge amount of fiber, and that fiber is collagen still. The same old fiber. It's just instead of liquid, squirting around through all of that, we primarily have solid material or a tough jelly-like material in the case of cartilage. And then blood, and there's cells scattered around in here. There's always cells scattered around that make the fibers, that maintain the connective tissue, but the cells are kind of a minority. Connective tissues have a relatively low amount of cells in them, so they're relatively cheap tissue to maintain. Epithelium is completely cells. This is just nothing but living cells, or dead cells on the outside surface, and living cells down below it, but cell, 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 cell. Whereas connective tissue is primarily not living material. In the case of blood, we've got a lot of liquid, and it's the only one where we don't have fibers, except when blood clots. When blood clots, we suddenly have a loose fibrous connective tissue. It looks a lot like these guys, and that's a blood clot. All right. So let's go ahead and take a look at the connective tissues right here. So here's an example. Here's some loose fibrous connective tissue. Here's the fibers, the little purple nuclei or nuclei of cells that produce this material. And then there's liquid in here. The liquid, like the liquid part of your blood, it really is just plasma that's leaked out into your body tissues. Whereas when we look at a dense fibrous connective tissue, like this bit right here, this is a piece of a tendon or a ligament. It's mostly fibers, a few cells supporting it, and a much smaller amount of liquid in here. Very, very tough, very, very strong. Cartilage, the fibers are very, very fine and very microscopic, very hard to see, and then they're all glued together with a bunch of jelly-like material. Imagine dissolving some cotton balls in some gelatin, like you're making Jell-O. And you heat up the Jell-O solution, you throw a bunch of cotton balls in there, and then let the stuff harden. Like Jell-O does. Well, that's basically what cartilage looks like. And then scattered about there are what are called chondrocytes, the cells that maintain the cartilage, regenerate the tissue, the fibers. They build the fibers and all of that sort of stuff. And that's cartilage right there. In bone, same sort of idea, but instead of settling on a gelatinized, sort of material, we in fact have this, solid calcium carbonate. There's other minerals in there as well, but calcium carbonate's the major one, making up the solid part of bone. And then all these dark lines represent channels full of fibers. The fiber is collagen. And then cells scattered about that maintain those. Typically, each one of these little dark dots has a single cell in it that maintains the bone around it in that location. So it's mostly non-living stuff, plenty of cells in there. And then, as you should know, in the case of your bony skeleton, quite a lot of blood supply and nervous tissue, supporting the entire bone right here. So it very much is a living organ, perfused, with nutrients and a blood supply and living cells and all of that. Adipose tissue is a particular type of loose fibrous connective tissue. The oil droplets here store energy very efficiently. Oil is a very, very high amount of energy that you can contain in a unit of volume and weight is higher than carbohydrates. So lipids are a very, very good long-term energy storage. And it turns out to be very useful as a thermal break between the outside world outside and the inside world of the organism inside. So subcutaneous adipose tissue, subcutaneous fat, is pretty typical. There's an example of blood that has not turned into a fibrous connective tissue. That's what will happen when these platelets are activated in a clotting event. All right. So we'll put muscle tissue and nervous tissue together because they're very closely related. Both of these are types of cells that can respond dramatically to an electrochemical stimulus. That means the term we use in physiology is they are excitable. An electrical charge can induce the cell to do something fairly dramatic. In the case of muscle tissues, that is going to be contraction. In the case of nervous tissue, like this neuron right here, if I stimulate this with an electrical charge appropriately, it's going to send an impulse all the way down the length of the neuron and release some neurotransmitters here at these axon terminals at the end where it's touching some other cell. And that other cell can be another neuron, or, as we very well know, that other cell can be a muscle cell. And so that nervous cell can stimulate this muscle cell to contract by an electrochemical stimulus. And what's going on inside muscle tissue, all of it, we'll talk about the differences here in a minute, is that they all have these contractile proteins. And classically, that's going to be actin and myosin. This is a highly specialized version of the actin that we find in microfilaments, and something kind of vaguely similar to the tubules that we find in microtubules inside all cells, all eukaryotic cells, the cytoskeleton. So muscle cells just have a highly, highly modified cytoskeleton that's arranged, so that the cell can change shape usually along one axis. It can shorten in length in one particular direction. That's the way muscle cells usually work. Now there's some differences between smooth, skeletal, and cardiac muscle cells. Smooth muscle cells tend to contract a bit more uniformly in all directions, while skeletal and cardiac muscle tissue, skeletal is stuff that we control our muscles with semi-consciously. We decide to move something. And cardiac muscle tissue, as you should know, is the muscle tissue that is unique to the heart of animals, not the circulatory system, just the heart. And these contract very much in a linear fashion. The two ends are drawn closely together to change the shape of the muscle quite dramatically. Smooth muscle you're going to find surrounding all of your hollow organs inside your body, including arteries and veins. And smooth muscle is responsible for peristaltic propulsion of material through your digestive system, controlling the diameter of the airways in your lungs, controlling the diameter of your arteries and veins to control blood flow, powering the movement of fluid out of glands like salivary glands or mammary glands, etc., etc. Cardiac muscle, as we said before, strictly for contraction of the heart, and skeletal muscle for conscious movement of your skeleton. Right. And the nervous system actually has quite a few different sorts of cells. As indicated right here, we've got a neuron, the main cell right here. We've got these oligodendrocytes, which provide an insulation around the axon of the neuron. So these are some extra cells that add to the function of the neuron. We've got some astrocytes right here, which is a type of structural and metabolically protective cell. It doesn't send messages back and forth in the same way that an axon does, but it helps keep the neuron with its axon alive. So you've got a whole lot of cells that are devoted to keeping a neuron alive, and those are what are called the glial cells. And there's about a 10-to-1 ratio of glial cells to neurons in the nervous system of any animal. Here's the term glial cells. All right. we want to talk a little bit about regulatory processes in animals. So all animals maintain something called homeostasis. All right, homeostasis. So homeo means the same, and stasis means value, right, or setting. And so, a homeostatic process that goes in us that we're all familiar with is maintaining our body temperature, right? We normally have not a perfectly fixed body temperature. It varies. It wanders around between day and night, and other stimuli as well. But it tends to stay narrowly between on either side of around 98.6 degrees Fahrenheit. And that is a homeostatically regulated mechanism. So homeostasis is regulated by negative feedback mechanisms, all right? So the classic example right here that's always used, we'll go on to this one in just a second, we're using food, but when you're talking about body temperature, what I suggested up here, if your body starts heating up too much, if your internal temperature starts going up, you're going to change some behaviors, or your body is going to do some things that will start pulling that temperature back down through negative feedback. So sweating causes more evaporation, which removes more heat from your body more effectively. More circulation going to your skin. Think about the elephant I was talking about earlier with its ears. You're making more blood flow through your skin by relaxing the smooth muscle surrounding vessels of arteries leading to the skin. And you can drop temperature. Conversely, if your temperature falls too low, you're going to cease perspiring. You're going to pull blood away from the external surfaces and keep it inside the body. And if necessary, you'll increase internal metabolism to generate enough heat to get back up to your set point temperature. And then those processes shut off. When we get back to the set point, negative feedback mechanisms shut off. They only turn on when we deviate from the set point, and they work to move back towards that set point. And that's what we mean by negative feedback. And that's how we maintain a homeostatic situation. So a negative feedback loop is simply the connection between the stimulus and the effector. So temperature goes down too low. Metabolism is triggered to, kick into higher gear. That's, metabolism goes up. More heat is generated. When the heat gets up to the set point right here, the sensor shuts off, stops stimulating your body to generate as much heat through basal metabolism, and that's the loop part. So the trigger, the sensor, the trigger, and the effector are all connected to each other in a loop. Here's an example using blood glucose right here. All right. Oops, I lost here we go, sorry. Here's an example of glucose regulation. And we're looking at just the function of insulin, but there's actually two hormones produced by your pancreas. That is insulin, which induces cells to take sugar out of the blood and store it away in the liver and store it away in body tissues in order to get the blood glucose levels back down where they need to be. There's actually another hormone, which I have looked up somewhere up here, which is glucagon, which is the antagonist to insulin. And if your blood sugars get too low in your tissues, in your blood supply, glucagon is released, and that causes your liver to release glucose back into your blood. So there's actually two hormones regulating blood sugar. One to keep blood sugar from getting too high, that's insulin, and one for keeping blood sugar from getting too low, and that is glucagon. And collectively, they both work via negative feedback loops to ensure that blood sugar levels stay within a narrow band. Because if your blood sugar levels get too high, you tend to draw more liquid via osmosis into the blood. That increases blood pressure, which increases stress and tension on the kidneys. It increases fluid content of your tissues because your circulatory system leaks liquid everywhere. So you have swelling and edema in the body tissues, and these are chronically very bad for you, long-term high blood pressure, and causes kidney disease and various other malfunctions, which are extremely undesirable and familiar to people who are familiar with the problem of diabetes. Too low a blood sugar level, which is controlled by another hormone called glucagon, is lethal. If your blood sugar levels get below a certain level, hypoglycemia, you'll go unconscious. If they stay too low, you will die. Your brain will not get enough, be able to generate enough ATP, and that will be the end. So it's quite a dangerous, lethal condition to have hypoglycemia beyond a certain critical level, and that's the role of glucagon, to regulate that, to keep blood sugar levels up to a certain level. So in the world of temperature control, thinking of your house, the first example I made, your air conditioner might be equivalent to insulin, trying to keep the heat down to a manageable level. A low level of heat, you can freeze to death quite deadly, and so that's the function of your heater, to keep temperature up, at a survivable level, and that might be equivalent to glucagon and its function to keep blood sugars up to a necessary level. And of course, when glucagon is functioning, insulin is not going to be functioning. That's over here. And when glucagon is exerting its influences, insulin is not going to be released, because they're both regulated by opposite ends of that feedback loop. Now, there are a number of things, processes in our body, that are controlled by positive feedback loops. They don't go on forever. They have a natural endpoint, but classically the muscular contractions of childbirth are regulated by positive feedback. And the difference between negative and positive feedback is that in positive feedback, the stimulus, which is stretching of smooth muscle in the uterus in this particular case right here, induces the release of a hormone called oxytocin, which, when released into the blood, causes smooth muscle to become more aggressively contractile. And when some smooth muscle making up the uterus contracts in some location, it necessarily stretches smooth muscle in some other location around here, because it's pulling on this, volume of a fixed volume inside the amniotic sac. And so that causes that tissue in turn to contract, which causes more oxytocin to be released. And the process builds and builds and builds until we create enough pressure right here, along with relaxing of the cervical muscles, which are not under the effect of oxytocin. They don't contract, they actually relax instead, while all of the rest of the uterine muscle is contracting, and we finally provide enough force to propel the embryo out of the uterus through the birth canal. And then that ends the positive feedback mechanism, because all of this stretched uterine muscle is now quite relaxed. None of it's being stretched anymore. And so that ceases the positive feedback process. Another example from nature of positive feedback is a rock fall or a snowfall on a steep mountain that dislodges more rock or more soil or more snow, and it builds and builds and builds. That's an avalanche. It's self-reinforcing by a positive feedback until, of course, it hits the valley floor and, it ceases necessarily. I would strongly advise you to watch this animation right here. All right, and then we'll go on and talk a little bit more about thermal regulation. So here's a great illustration of thermal regulation in animals. Notice these are penguins, which are endotherms, warm-blooded vertebrates living in the southern hemisphere, Antarctica typically, but also the southern bits of Africa, South America, islands off the coast of Australia, and a few other southern islands. By the way, there's no penguins in the northern hemisphere. They're a strictly southern hemisphere animal. All right. So we defined endotherms and ectotherms before, but we'll just double check that again. So an endotherm generates its temperature internally. Ectotherms can regulate their temperature as well, but that can only be using the environment around them. So this is why snakes like to sit on hot rocks and hot roads at night, because they've gone there to counter the cool air by sitting on a warm surface to gather up heat energy. And classically, a lot of snakes hunt at night because they have a better chance of ambushing their prey but they'll sit on those rocks and those hot roads in order to maintain the high body temperature needed for rapid muscle contraction to be able to chase their prey. So that's why that behavior is going on. Now the flip side, endotherms maintain their temperature continuously, so they're always ready for action. They're always at full temperature. They're always at optimum functioning temperature. So no matter what's going on in the environment around them, ectotherms have to get that energy from their environment. So if it gets too cool everywhere, they cease functioning. However, since they don't maintain an internal body temperature and they have a very low basal metabolism, they need very, very little energy when they are at low body temperature. Whereas an endotherm in cold weather needs ever more energy to maintain its body temperature in cold weather. So the colder it gets, the more expensive the endotherm is to run. The colder it gets, the cheaper the ectotherm is, although the ectotherm gets to a point where it can no longer hunt for food. So it can only stay cold until it uses up all of its stored energy, in which case it will die. And that's why ectothermic animals don't typically live at the coldest extremes on the planet. The last places you'll find animals on Earth, those animals are going to be endotherms, able to, in air, not water. I talked about water before. But in exposed to the air, they're going to be endothermic animals. So up in Alaska, you don't find a lot of reptiles. You don't find a lot of insects, except lots of flies and lots of mosquitoes, which breed quickly during the warm temperature. But in general, not a lot of invertebrates in those environments. Control of temperature, heat conservation, and heat dissipation. I talked about that a little bit. Small animals that generate a huge amount of body heat, like a hummingbird, have to have an awful lot of means of getting rid of that excess heat. Body shape and coloration and things like that all allow for animals to thermoregulate. So if you want to get rid of heat, you need to increase your surface area and get more body heat to that surface area. If you want to conserve heat, you want to minimize surface area. So think about rabbits. They all maintain roughly the same internal body temperature. Rabbits that live in incredibly cold areas, like Arctic rabbits and Arctic hares, have very short legs, thick cylindrical bodies, and very short ears. That's to reduce surface area to hold heat in a very, very challenging cold environment. Whereas hares and jackrabbits and rabbits that live in really, really hot environments have very slight, elongated bodies, long legs, very long ears, and all of that is so they can dump heat into their environment more effectively. Additionally, I talked about adipose tissue. We have two types of adipose tissue in the world of animals. There is what's called white fat, which just means, when you look at it, it's a relatively pale, whitish, yellowish sort of color. And that is strictly a passive sort of adipose tissue that functions as a heat block and as an energy storage mechanism. There's also a type of internal adipose tissue called brown fat that we have in our body cavities that is specialized. It's brown because of all of the hemoglobin and myoglobin glycogen that you find in the tissue right there. And these cells have a huge amount of mitochondria that oxidize sugars very, very rapidly that are made from the lipids that are stored in that brown fat, and they can generate a huge amount of body heat. Those are the cells primarily responsible for internal generation of heat in animals. Neural control of thermal regulation. So your nervous system is the ultimate regulator of internal body temperature through an organ in your brain, the hypothalamus specifically, where the temperature of blood triggers cells that in turn control various parts of the nervous system that regulate blood flow, sweat production, and all of those sorts of things through negative feedback loops. All right, so there's a fair amount of terminology right here. As a general rule, you want to go ahead and get all of these terms into your vocabulary. Some of it will be in the test as it's relevant to any particular topic. look them up. There's definitions right here. You can always look them up, to get a better understanding of them. I would strongly suggest that you go ahead and go through your summaries right here. Go back and read the material. Try and answer these questions. Here's a series of questions right here. Test yourself with this material. In addition, I'll post a graded quiz in Blackboard that covers the same material. Here's some open-ended questions. If you can answer these questions, then you've prepared yourself fairly well for the test of this material right here. And that includes this recorded lecture.